We could get Dr. McFarland and Dr. Bartenschlager to join us here at the table. We can have 15 minutes or so for questions now. So while people are running to the mics, I can start off. So Sonia, normally how many cells do you sequence from an individual, and have you had a chance yet to sequence across similar cells across multiple individuals?
So that's an excellent question. So we sequence per individual, we target 10,000 healthy cells. We really usually come out with about 5,000, but we sequence all the dead cells as well. So what I didn't show is that we use gating post acquisition of the data to remove those dead cells. And the reason that we do that is because, as immunologists, we want to have our sample as enriched for healthy cells as possible. We want to clean them up. We want to use beads. We want to use sorting. But all of those steps actually really impact the very sensitive cells, like the endothelial cells and the hepatocytes that have been dissociated. The T cells and the macrophages, they survive a bit better. But in order to see all the cells, including the more fragile cells, we really need to, we really kind of took everything and then used bioinformatics to, using the cells that are expressing very few genes, we just gated those out.
Okay, we'll start with questions here.
I have just a quick question for Ralph, actually. I was curious about the LGP2 protein. It's sort of been controversial whether it's sort of antiviral or it dampens the antiviral response. And so you showed really elegantly that it seems to be dependent on RIG-I, I think, right? And so does it seem that it's able to, the response is really promoting the antiviral response, or does it seem to have this sort of negative phenotype?
Actually, it has two different phenotypes depending on what the pattern recognition receptor is. So in the case of RIG-I, we do see an inhibition, but in the case of MDA5, we do see an activation. And the activation we see fits to MDA5 being the sensor for delta and for hep C, whereas in the case of, let's say, flavies like dengue or Zika, it's RIG-I sensing, and that's the opposite. So it has really two different roles. We have kind of an idea how that works molecularly, but it's just an idea so far. But it has two functions.
Hi, my question is also for Dr. Bartenschlager. So wondering for the MAVS protein, you showed that it's required for mounting the interferon response against HCV infection. I was wondering, are there any studies looking at overexpression of MAVS, either the wild type or the uncleavable form, and whether or not this confers HCV resistance to infection or a more robust clearing of the virus?
Well, as I said, so when you have non-cleavable MAVs in the cells, the virus is better controlled as compared to when it can cleave MAVs. So we think the virus is pretty good in controlling the MAVS pathway, but it's very lousy in controlling the TLR3. And in fact, we never saw cleavage of TRIF, what has been published. And I think the virus is poor in controlling TRIF and thereby TLR3. So my guess would be in cells, also including hepatocytes, TLR3 plays an important role with the ISG signature that we see in infected livers.
Sonia, that was a wonderful presentation. I was curious, some groups have been able to do West Nile inclusive single-cell RNA-seq. I was wondering if there's a means to get hep C signal from this, or is there an issue of RNA abundance in these hepatocytes?
So we haven't looked at that yet. We've looked at it in the woodchuck, and we can see some transcripts for core and for protease. We haven't done that yet, but we will.
Great.
Yeah.
Hi, Sonia. Really elegant presentation, very beautiful data. So I was wondering, Based on some of the location of the cells data that you had, we didn't dwell too much on it. I wondered if you could speculate what cells might be involved in influencing the cells that are circulating through the liver. So as the blood feeds the liver, the pressure slows, the flow slows, and so there's this opportunity for either environmental influence or cell-cell contact. And you sort of mentioned some of the cell types that you thought were close to those areas in health and how that might change in a diseased liver.
Yeah, that's a great question. So what we're seeing, actually, we're doing some EM from some of the HCC samples, and what you're seeing is that those endothelial cells that are nicely lining those sinusoids are actually broken. So that barrier is broken. So the endothelial cells would kind of protect the immune cells that are going through from some of the things that are, that are taking place in the hepatocytes. So the endothelial cells will probably interact a lot with the immune cells, but also the cholangiocytes and the bile ducts are very close too. So if there's any kind of disruption of those different cells, then they would probably have a role.
Okay, so we have some more time. Ralph, have you looked at LGP2 in the context of other viral infections?
So far, as I said, so far we have looked in the context of Delta, and there definitely it's behaving pretty much like for HCV. For Flavius, we took a look at. That's what we have around in the lab. It's very convenient. For Hep B, unfortunately, there's nothing you can look at because, at least in our hand, this virus is really flying under the radar and there's pretty much no induction whatsoever. It's very interesting. You can have Hep B and Hep C in the very same cell, and when you treat with interferon, HPV doesn't care, but HCV is gone. So it's really, they co-replicate quite well. They coexist, and one is highly susceptible, the other not, which to me tells HPV doesn't mess up with anything of the check stat signaling or so, because otherwise HCV wouldn't be eliminated. So coming back to LGP2, we looked for some hepatitis viruses, B and D, and Flavius, and I would say to me the most striking was the real similarity between Delta and Hep C, how they are sensed by MDA5 and LGP2.
So I have a question for Ralph. I might have missed it. You said that the HDV can be enveloped by HCV. So is there a potential the HDV co-infection could exacerbate the HCV infections?
That's an interesting analogical consequence of this paper that just came out. It was a study from François Licouset in Lyon. And they could show, at least by pseudotyping, that you can have a delta that is enveloped, for instance, by HCV and other viruses. So then, of course, the question is, from an evolutionary point of view, why is delta so addicted to Hep B in vivo and not to others? Maybe we just didn't look for that. And that's why we didn't see it so far.
If I may, one question for each speaker. First to Dr. Martin Schlager. I'm very puzzled by these naked viruses that exist in fishes. And actually relating to hepatitis D or Delta virus, it's kind of a naked virus, but I was always under the impression that it existed or it evolved because of the presence of HPV. So there are kind of two questions that come out of my head. One is, how do these naked viruses get transmitted? And second, was HDV present before HBV, or it really came after? So you're just mixing my head with evolution of viruses.
So we are equally mixed. For these Naketna viruses, actually, we don't know how they are transmitted between fish species. The point is they are pretty abundant in different tissues, so they do not have this liver tropism as we see it for Hep B. And our thinking is simply with the acquisition of the envelope that is really specifically binding then to NTCP on the hepatocytes, there the virus underwent a restriction to become hepatotropic, which is not the case for the Naketna viruses. For the Delta virus, where it came from, this was always the big question, because this circular single-strand RNA with this rod-like structure, as we see it for Delta, they are pretty abundant in plants. And then we have this real outlier HDV in humans. So isn't there anything in between? And the answer is yes, it is. And we just didn't see it before because we did not have the power of all the sequencing technologies. And the fact that this can be enveloped by different viruses, I think we really have to totally reconsider the evolutionary history of Delta virus and how this developed. I think there's probably way more out on Delta virus and Delta-like viruses, and whether or not they are also pseudotyped by other viruses or whether they have other ways of transmission. I think this is completely open for now. But I would say this is a really exciting time to go also in these evolutionary aspects and consider where did these viruses come from, how did they co-evolve, why are they so well adapted to their host, and what is actually their transmission as they have evolved across the different species. So for now, we are equally mixed as you are.
Thank you. And Sonia, if I may, so it's really fantastic work that you've been accomplishing. And I'm a little bit more skewed toward the hepatocyte populations. So right now, you can identify subtypes. So do you think that in the near future, you'll be able actually to capture this population and then study it before doing the RNA-seq analysis so that you can actually change the phenotype ex vivo after you isolated the cells?
So the tricky thing about the hepatocytes that you know very well is that they're very hard to culture. So one of the things that we're trying to do is we're trying to do precision cut liver slicing and culturing the hepatocytes in transwells in that liquid air interface, and then we can use single cell RNA sequencing to honestly screen the different types of drugs that would and so one of the ways that we can do this is by using bioinformatics to repurpose drugs that are already available to treat the different hepatocyte populations. Or the other thing we can do is read out the hepatocyte function, but treat the slices with different immune populations we pull out from the map. So what we're trying to do right now is, for example, take the immunoregulatory macrophage populations and embed them in the slice once it comes out. Because we know in general, if you have a slice and you culture that for 7 days, it starts to become fibrotic naturally. Right, so you could pull. What you can do is pull out those macrophages and put them in that slice and see how it impairs or changes that path to fibrosis.
Great, thanks.
Excellent talks, both of them. I just wanted to ask Tanya, have you tried to culture any of your hepatocytes in human serum or in fetal bovine serum?
Great.
Because there's quite a difference in our labs.
Yes, absolutely. So we've cultured them in human serum. We've tried a little bit of knockout serum, but we're trying to stay away from bovine serum. But one of the things that has worked actually quite nicely is trying to culture the hepatocytes as organoids rather than culturing them in 2D.
So a question for Dr. Bartenschlager. So not that I'm suggesting you become a snake and fish immunologist or any other kind of innate immunologist. It's interesting because snakes nor salmon have the later type 3 interferon response. It's a kind of higher vertebrate sort of thing. And it's interesting. Do you think it's really difficult in higher vertebrates where the type 1 and 3 interferon pathways kind of converge? Do you think there's an opportunity using some of the more naked viruses and or HDV to dissect out what the exact role of lambda or other type 3 interferons might be?
That's actually a good question. We have started a bit along those lines to look, for instance, how evolutionary conserved are some of the features, like, let's say, targeting of signaling molecules like MOFs and so on. I have to admit, I have absolutely no clue about the immune system of fish and how they actually handle pathogens. So we have just organized to get some fish culture, or fish cell lines. It takes ages because they grow at room temperature. So, but this is, I would say, a really interesting question. How conserved are those features across, let's say, these kind of viruses from fish up to humans? It might be that they could be used if we know a bit more about them. But as I said, we have first to do a little bit of a comparison between fish and human before we can make a conclusion about that. My biggest hope would be that at some point we find hepatitis C in mice, but ideally lab mice, not wild mice. Then we would have a mouse model, or to use some of those which Charlie Rice had already done with this red Hep C as a surrogate. I think that's the spectrum of these viruses offers new possibilities to look into that.
Certainly the role of type 3 interferons for liver viruses and other infections is
Correct.
interesting. Big trial back in the day with Hep C, and just an interesting thing.
So we
don't
know
much about.
it's a fantastic marker, and we have no clue why.
Okay, so I'd like to thank the speakers for 2 excellent talks. You guys can